So each contestant will have about 12 minutes to present and your time will start at the second you speak. I'll give you 1 minute warning by interrupting your talk over Zoom. Uh, so before you finish, I'll give you 1 minute warning. Um, Once you are ready to present, give me some verbal cue like start, and then I can advance the slide dock. And during the presentation also, I'll be navigating the slide dock. So please give me a verbal cue like next, advance, or proceed, so I can advance to the next slide. I'll be introducing you before your talk, so you can directly jump to your presentation and save your time. And at the end of the presentation, judges can unmute themselves and ask questions to the participants for about 5 minutes. And while judges are scoring, I'll moderate the question from general audience, and I ask the audience to uh, mute themselves, stay muted during the presentation, and stay muted during and after the presentation, and drop their questions in the chat. And after the presentation, then I can direct their questions um while the judges are scoring. So these are the ground rules we have. So uh, if everyone's okay with that, then we can move to our first presenter. Roya, are you ready?
Yes, sure.
Okay. So let me introduce our first presenter, Roya Bagri, from Mechanical Engineering and Engineering Mechanics Department. And her presentation is Electroconductive 3D Carbon Nanotube Forest Scaffold for uh, Cardiomyocyte Maturation. So if you are ready, we can start.
Okay, start. Hi, everybody. As you can see, these are the headlines for my presentation. Next. Cardiovascular diseases are the leading cause of death worldwide. So as you can see in the chart on the right, these are the leading causes of death in 2020. Also, still, there are many cases for COVID-19. Still, the heart diseases is the main lead for deaths in the United States. Near to 70.9 million people dies from this disease each year. And the estimation is that the near to $1 billion will be the cost of CVD in near future in United States. Next. There are different treatments for cardiac repairs rather than surgery. As you can see, pharmacology, which use the medicine, cell therapy, injection of cells, and tissue engineering, which is the combination of used cells, biomaterials, and the engineering principles. So the cell therapy and tissue engineering both has the purpose to develop the mature cells. So today I will focus on the tissue engineering to use a scaffold and biomaterial to produce the mature cells. Next, please. Can you play next again? So in heart, one of the main properties of the heart is the electroconductivity of the heart, which cause an heartbeat. So the reason for this heartbeat is the sinoatrial or SA nodes in heart, which these are the cluster of cells, and they applies and impulse the electrical impulse to the myocardium, which are the cardiac cells. So to mimic these properties of the heart, we want to use the carbon nanotube forest or CNT forest as a scaffold or a biomaterial for cardiac regeneration, as it has a mechanical and electroconductivity properties, a noticeable one. The carbon nanotube are rolled up graphene shields, and the CNT forests are the grown vertically CNTs together, which represent as a forest. Next. We use this carbon nanotube forest in this project to produce a mature and functional cardiomyocyte, and also we can use this cardiac patch as an in vitro model for heart muscle tissue for drug testing. Next. Okay, so for preparation as scaffold, for CNT forest as scaffold, first we will use the alumina and iron catalyst nanomaterials on the silicon wafer to help the nanostructure of the scaffold. Next. After that, in the condition of 750 degrees centigrade, with the presence of ethylene, argon, and hydrogen for 12 minutes, we grow the CNT forest. Next. The CNT forest growed wavely and entangled with the average heights of 200 microns. Next. After preparing the CNT forest, the CNT forests were sterilized with 70% ethanol, and it was under UV exposure for overnight. Next. The CNT forest was coated with 1% gelatin for enhancing the adhesion of the cells on surface of the scaffold. Next. What happened was after preparing the scaffold, the isolated cardiomyocyte, which we extracted from neonatal rats height 1 to 3 day old, and we see that on the CNT forest. Next. So you can see the morphology of a scaffold, CNT forest, a scaffold here before and after the coating. So you can see the side view and top view. After the coating, the CNT were bent, and they created some island on top of the CNT forest, which are the reason of electrocapillary force of the gelatin in the coating. Next. For physical characterization, conductivity and impedance, the CNT forest was, the conductivity was measured for CNT forest in PBS, and coated CNT forest in PBS, and it shows that there was no significant difference between them. It shows CNT forest has a conductivity in liquid. The impedance, the electroconductivity resistance was a measure, and it shows that after the coating, the impedance was decreased because of the presence of gelatin coating. Next. Here you can see the schematic of the cardiomyocyte in this 3D CNT network. On top, you can see the side view, and after the coating, the CNT were bended, and the cardiomyocyte was performed in top and between in the side walls of the island. And in the top view, you can see the presence of island and the cardiomyocyte on top of them. The SEM pictures on the bottom show are the proof for this schematic. And the left side, you can see in this side walls of the island and on top of the island. Next. To study the viability, the live dead assay was performed. You can see the images of live dead pictures. Live cells were labeled as green, and dead ones are labeled as red ones, and the viability was calculated by counting the live and dead cells. There was no significant difference between CNT control, which was showing that the CNT force has no toxicity. Next. Another viability test that we measured was the press-to-blue assay, which represent the absorbance of the number of dead cells here in different culture days. You can see that there were no significant difference between the CNT and the controls for each days, which is the proof for another non-toxicity of the scaffold. Next. To visualize the cardiomyocyte formation on the scaffold, scaffold, we used different cardiac markers, troponin D, alpha-actinin, and DAPI, which represent the nucleus, troponin C, which represent the cardiac muscles, and alpha-actinin, which represent the cytoskeleton in the, you can see the elongate cardiomyocyte in day 14, which shows the mature cardiomyocyte on the scaffold. Next. The adherent cells, well-adherent cells on top of the island, and in well-adherent cells on side walls of the island in different magnification, you can see here. Next. Also, we applied the electrical stimulation to our samples because we wanted to improve the maturation of the cardiomyocytes. We used the customized chamber, as you can see in the picture. The samples were seated between the two carbon rose electrodes, and it was connected to the voltage source. It was applied for 3 days in the incubator in the medium culture. Next. The result of the gene expression after the electrical stimulation, you can see in this slide, we measure different cardiac genes here, actinin and troponin T, which already we visualized in previous slide, and a GGA, which is for gap junction and for the connexin gap junction, and MYH6, which is for myocardiac muscles and cell. In all of the cardiac genes, it shows that the CNT4S with stimulation has a significant difference with other samples. And also in troponin T, it shows that the controls with a stimulation and non-stimulation has a significant difference. The sum of this gene expression result was that the electrical stimulation caused in a more mature cardiomyocyte on top of the scaffold. Next. In conclusion, so we prepared an electroconductive scaffold, CNT4S coated gelatin scaffold, we seated cardiomyocyte on them. The cardiomyocyte well adhered on top of that, and there were no sign of toxicity after applying the electrical stimulation after 3 continuous days. It shows that the cardiac genes shows the enhanced, which was representing of more mature cells. To sum up, we can see that the CNT4S can be an ideal candidate for cardiac muscle regeneration and can be effective for mature cardiomyocyte. Next. And you can see the acknowledgement here, which was funded by American Heart Association.
Can we ask questions now?
Yeah, that now judges can unmute themselves and ask the questions.
All right. Yeah, great talk. I have 2 quick questions. One is that if you have, so like I maybe missed the part, but the cells you kind of seeded is it like iPS derived cardiomyocytes or is it neonatal ones? Second question is that, I see that, that there's a single cells you imaged with the SEM. Are you also interested in to kind of seed more cells to have like more having the multicellular, network or monolayer of these cardiomyocytes instead of just patches of the single cells?
So your first question, our sample was isolated from neonatal rat's heart.
Okay.
1 to 3 day old. And the other one, it's like, so there were some number of cells on top of the island, but as you can see in the, if you go back to a slide next, sorry. To a slide.
No, this one?
No, next one.
2 slides.
No, I mean, next, not
before.
Yeah. And next, sorry. If you go next. sorry, it's really hard this way. You can see the number of cells on top. It's after fixation and this kind of process, so the number of cells would decrease a little bit. But if I get your question correct, you were just saying that we put like 2 layers of CNT on top of each other with the
No,
cell.
no. I'm talking about the confluent monolayer. Like instead
of cells.
they are kind of right now they are detached from each other right now. There are some gaps in between the cells.
Yeah, that's correct.
So it's not like tissue-like yet.
Yeah, so we can use more number of cells for that, or we can use like some growth factor to help the growing of the cells. But for now, our project, it was just like based on this.
Mm-hmm. And what is this? What does this CS mean?
It's CNT. It's carbon nanotube forest.
Yeah, CS.
CS. Oh, it's a cover slip. It's a
Cover slip. I see. Yeah, you didn't talk about it. Okay.
Oh, sorry, I said control. I'm sorry about it.
Yeah. Okay.
All right, very nice talk. I'm a fruit fly biologist, so this is a little bit, I'm going to ask like the big picture question. So you say, okay, you grow these cells in a forest, but I don't know, what have people done before that, and why? What is the significance of growing them in the CNT forest? I mean, I saw electroconductivity and things grow, things express genes, but is this growing in a forest like enabling you for the first time to grow these cells? Question number one, because what was before you grew them in this forest? There must be a reason why you grew them in the forest. And number two, how far are you away to actually use these cells in patients in the long term, like big picture? You start growing them in a forest, then what will come after? You mentioned a little bit in the beginning that you can inject cells, right? Heart cells, and they will repair the heart. So if we can do that, how does the forest come into place? That just to multiply them to have enough to inject? Or so that's kind of my question.
Yeah, there were some studies around the CNT before that, but the specific properties that CNT forest has because of its morphology and like the electroconductive and mechanical properties was another reason that we use that. So for that, the other reasons, like the purpose, I mean, the idea is like, if we can see that we can just extract the cells from cardiomyocyte and freeze them and then inject them into the patient, but we are not still in that step for the project. But all of the purpose of this scaffold is to make the cardiomyocyte, I mean, enhance the maturation of the cardiomyocyte because of the electroconductivity that the CNT forest has.
Okay, thanks.
Okay, then till judges are scoring, I can let the general audience ask question if they have. And judges, please let me know when you are ready with the scores. Give me a nod or something, then I'll know.
So what, excuse me, what do we do with the scores? Do we submit them online somewhere, or are we going to tell you the scores?
No, you should submit them online.
I did not see the submission. I saw a Google Drive folder, but I didn't see anything where they can be submitted in what I got since yesterday night.
You can upload it to the Google Drive folder. I'll get back.
Yeah, there is a Google form there.
Yeah.
Score sheet for GRC 2021.
Is that, sorry, I don't want to hold this up. Oh, the copy of Judge Sheet 2021 docx. But there's nothing. There's no way I can put them.
Yeah, not that one.
Document is
form. Yep.
Google form. Document is for the
copy for Judge. Sorry, I'm completely lost in this. Google is so disorganized. I don't think I have it.
So in the email about the packet, it's not included as attachments. It's just in the hyperlink there.
Yeah, I have that, and I clicked through everything. I just don't see anywhere where I can put in my scores.
I'll send you the scores then.
Okay.
Quickly.
Yeah.
So
thank
if you can note down them, somewhere, then I'll send you. Then we can at towards the end we can do that.
Okay, sounds good. Yeah, I don't want to keep you guys. Yeah.
So
Alright.
I just want to mention something. The schematic and some picture were not exactly what they should be. I think after downloading them, it was a little bit changed. It was, yeah, I just want to mention that.
Um,
If you go back in the schematic, it was not supposed to be this way. It's like a cubic thing. Um,
which slide?
if you go back for a schematic preparation parts, yeah, I think, yeah, go back
This one.
two more. For this one, they are not supposed to be like this. They will like be placed together. And I think it's because you use the, I don't know why, but the one that I have, they are like placed on top of each other. So it's like a tree. Yeah.
Um, okay, I think probably because
No, I
it's
just want to mention that.
yeah. So I think now it's the time for the next contestant, and for the general audience, please put your questions in the chat so we can address those questions towards the end. Okay, so let's get started with the next contestant. Um, so the next contestant is Meena Chandrasekhar. Meena, are you there? Can you?
Yeah.
Okay. And she's from the Department of Chemical Engineering, and her topic is high solid roller bottle, enzymatic hydrolysis, and respirometer fermentation for screening lignocellulosic biomass. Shall I start the presentation?
Yes.
Meena, are you ready? Okay.
Yes. The next slide. Lignocellulosic biomass is a valuable commodity as an alternative to petroleum for the production of biofuels. Agricultural residue like bagasse, food crops like sugarcane and corn, and wood-based feedstock like poplar and willow, and certain domestic and industrial waste materials are some examples of lignocellulosic biomass. Next. Lignocellulosic biomass is composed of an intertwined network of lignin, cellulose, and hemicellulose. It is essential to break down this complex network in order to convert it to sugar molecules. A typical process to convert biomass to ethanol consists of three main steps: pretreatment, enzymatic hydrolysis, and fermentation, followed by product recovery. Pretreatment is carried out by applying a combination of heat and pressure to separate cellulose from hemicellulose and lignin. During enzymatic hydrolysis, cellulose and any hemicellulose that is remaining after the pretreatment are decomposed to monomeric sugars, which is mostly glucose and xylose. The monomeric sugars are then fermented using yeast or bacteria to produce ethanol. Pretreatment methods, engineering enzymes, and genetic modification of fermentation microorganisms have been extensively studied in the last few decades. Next. Next. In the recent times, focus of many researchers have been on identifying the environmental factors on lignocellulosic biomass. Abiotic factors like water availability, drought conditions, soil nutrients, etc., play a major role in the feedstock yield and productivity. Different cultivation experiments are being carried out for this purpose. Usually, these experiments yield a limited quantities of biomass, which is less than 1 kilogram. Bioreactors are usually not designed for such small quantities. Therefore, it was essential to devise a small-scale enzymatic hydrolysis method which can process multiple samples in small quantities. Next. Most studies focus on unprocessed feedstock composition or sugar yields after hydrolysis. However, there are inhibitory compounds which are generated during the pretreatment which hinder fermentation. Very few studies talk about the fermentability of hydrolysis, or they are limited to predicted yields. In our study, we conduct enzymatic hydrolysis and fermentation experiments to identify the effects of environmental factors on feedstock. Next. Next. High solids loading condition. Next. 20 to 30% solids proportion by weight can be considered as high solids loading in enzymatic hydrolysis. Why do we need such high concentrations? It is because that this is the industrial requirement for fermentation experiments. Next. However, there are some bottlenecks in high solids loading. Unlike bioreactors in industries, which have complex mixing arrangements, benchtop scale experiments have to address bottlenecks like low water availability and high viscosity. Low water availability and a high viscosity of the biomass leads to mixing issues, which in turn will lead to low sugar yields. As you can see on the image on the right, you can see that there is absolutely no water available for the biomass to disintegrate. Next. Currently, lab experiments involving high solids loading are carried out in shake flasks. The mixing and low water availability issues are resolved by using the fed batch mode. In the fed batch mode, the biomass is added to the flask in smaller proportions at different times. However, there are some issues with the fed batch mode, which is it requires more space and the solids get accumulated to the walls of the flask and then they're resulting in low sugar yields. Next. In the roller bottle method, we conducted experiments in centrifuge tubes which were placed on the horizontal rollers as shown in the picture. The solids loading that we tested are 6% and 9% glucan loading, which were about 20 to 30% solids by weight. And the fermentation experiments were carried out using baker's yeast, which is Saccharomyces, and a commonly used bacterium, which is Zymomonas. We tested this method for various feedstocks, which includes corn stovers, switchgrass, sorghum, miscanthus, and mixed prairie. Next. In this method, we optimized the parameters like solids loading, phosphate buffer concentration and pH, and the centrifugation time. So the biomass was previously pre-treated using A6 pre-treatment method, and then this biomass was added to the centrifuge tubes, 85 ml centrifuge tube, and then water and the phosphate buffer required for that quantity was added to it, followed by autoclaving. Autoclaving is done to inhibit microbial contamination, and then enzymes were added to the mixture, and which were in turn placed on the roller bottle apparatus. This roller bottle apparatus was placed inside an incubator at 50 degrees Celsius. The experiment was carried out for 72 hours. And after 72 hours, the samples were centrifuged at 12,000 RPM for 3 hours, and followed by a two-step filtration. This filtration is done to separate the solids from the liquid fraction and also to sterilize the same for fermentation experiments. After filtration, the pH of the hydrolysates were adjusted to 5.8, which is a typical requirement for the fermentation microbes. Next. In both of the graphs, we can see that xylose and glucose concentrations are higher for the 9% glucan loading. This is simply because there was more biomass to begin with. However, when we compare the xylose and glucose conversion with respect to the sugars that were in the raw material, that is the untreated biomass, we can clearly observe that the conversion is higher for the 6% glucan loading. This is happening due to many reasons, including higher sugar concentration, which limits the hydrolysis process, which is very typical at extremely high solids loading. That is what is happening at the 9% glucan loading condition. Next. Next. Hydrolysate volume was tested for different buffer concentrations and centrifugation times. The 6% glucan loading samples had higher filtrate volumes. At 9%, there was no hydrolysate at 1 hour centrifugation. That is why we don't have any data points for that 1 hour centrifugation for 9% glucan loading. The volumes of hydrolysates were higher for 6% glucan loading at all conditions, which makes it evident that the liquefaction was better at 6%. Next. The final process parameters are 6% glucan loading with a phosphate buffer pH at 3.0 and the buffer concentration was 0.1 molar, and the centrifugation time was taken to be 3 hours. Next. Comparing this with the shake flask method, we went on to compare the results with shake flask method for the same experimental parameters. We performed batch mode shake flask experiments in a shaker incubator for 72 hours at 6% glucan loading. The biggest, the next, the biggest difference we found was hydrolysate volume. The roller bottle method had higher hydrolysate volumes compared to the shake flask method. The liquefaction was better, which can be explained by comparing the hydrolysate volume with the initial working volume of the experiment. For the shake flask, the working volume was 50 ml, whereas for the roller bottle, the working volume was 35 ml. Based on the hydrolysate volumes, we can infer that the liquefaction in the roller bottle method was 40 to 50% higher than the shake flask method. Next. Better liquefaction translates to higher glucose and xylose yields, uh, which were 42 to 47% higher for both xylose and glucose for the roller bottle samples. Next. Fermentation experiments were carried out at the University of Wisconsin-Madison. The picture on the right shows the setup for the spirometer experiment. Overnight cultures of Zymomonas and Saccharomyces were inoculated into serum bottles, as shown in the picture. At 30 degrees, the experiment was carried out for 48 hours. Fermentation processes usually release carbon dioxide, and the volume of carbon dioxide has been used as a surrogate for the ethanol performance. Next. The roller bottle hydrolysate and fermentation results were compared to one of our previously studied switchgrass samples. The switchgrass harvested during the Midwestern drought year, which is 2012, showed inhibition to the growth of Saccharomyces due to the inhibitory compounds produced in Apex pretreatment. All of these experiments were carried out in a 5-liter bioreactor. Similar results were obtained when the roller bottle hydrolysates were fermented. This makes our results consistent with our previous work. Next. This is a graph which compares the carbon dioxide produced during the fermentation experiment. 2010 switchgrass is our control sample, and we can see that in the 2012 switchgrass, there is almost very little volume of carbon dioxide that is produced, except for one sample, which was an outlier. And this shows that our results are consistent with the previous study. Next. To summarize, the roller bottle method can be used for screening a wide range of feedstocks. As a standalone method, we can utilize this setup to test a variety of pretreatment and enzymatic hydrolysis process conditions. We have obtained
One minute
higher
left.
better higher sugar yields and better liquefaction when compared to the shake flask method. High throughput in the roller bottle method has an advantage that the space constraint in the shake flask method is overcome in this method. That concludes my presentation. I would like to thank GSG for the wonderful opportunity. Thank you all for your time and attention.
Okay. Thanks, Meena. And I think now judges can unmute themselves and ask the questions.
A very nice talk. Yeah, Dr. Torner, do you want to go first?
Yeah, I want to go first. I kind of lost, it was a lot of data. I liked your talk and I kind of got it, but I'm outside of the field. Is there any take-home message? It was like data, and I was trying to keep up with you. Is there anything you can give me to take home, the take-home message? What is the big conclusion, the big picture of this?
So ultimately, the research towards biofuel production optimization is going in different directions, like optimizing the process conditions for pretreatment, enzymatic hydrolysis, and fermentation. These are separate studies. However, there are fewer studies which focus on the cultivation parameters like soil conditions, water availability, and everything. So the big picture is that the GLBRC has cultivation experiments in which we have about 200 to 250 samples of different plant materials that can be produced in the US. And to test such a huge amount of samples, we needed a method that can process quickly. So that is the use of the method that we have devised.
That makes sense. Thank you. Thanks.
Welcome.
Yeah, so follow-up question is that the main method you use is this roller-based method, and that is better than the shake flask method. I'm interested in why do you think that roller method is makes better fermentation and other type of this biofuel synthesization? Synthesis?
So
Yeah.
the first and foremost reason is that in a shake flask method, the mixing is not happening properly. So it happens in an Erlenmeyer flask, and the particles, as the experiment proceeds, these experiments are usually carried out for about 72 hours. As the experiment proceeds, the biomass gets stuck to the walls of the flask. This bottleneck is overcome while we are putting the samples on a roller because horizontal mixing is taking place there. In horizontal mixing, the accumulation of the samples to the walls are reduced. So this reduces the bottleneck. And another major reason is most bioreactors have horizontal mixing. So our method is closer to the industrial conditions.
Mm. Is there, do you think you can, so what you say is pretty makes sense, I think. Is there any, like computational simulation, CFD work that can kind of support that, your conceptual explanation?
We have not performed a CFD analysis for this method, but in particular, but there are different studies and theoretical explanations for how horizontal mixing works better, and that is primarily why it has been used in the industry over the other way of doing it, the shake flask way of doing it.
Yeah. So yeah, the industry, so like since industry uses it, it should be better that it's not academic, reasoning, right? So you want to show it, right? You want to kind of say the why in terms of the maybe your fluid mechanics or other types of the theoretical reasoning.
Yes, we are not focused on the fluid mechanics part of that for this study because the bigger picture is to uh, screen the samples that are produced in the plant cultivation experiment. But however, our lab is again focusing on to identify the the fluid mechanics.
Yeah. All right, thank you. Yeah, your work is great. I think it's very promising. Yes.
Thank you.
So now it's time for any questions from the audience. Please put your questions in the chat, and meanwhile, judges put the score for the presenter. And please let me know when the judges are ready after the scoring. So meanwhile, we are waiting. Meena, I have another question as well.
Sure.
So do you plan to extend your research for some areas? I think probably you have uh, done this for the US in the United States, right? So are you planning to extend in some way?
Uh, so I don't think we there are different pretreatment methods that we work with in our lab. This method is promising to use like two or three of those methods to classify the feedstocks based on the yield. However, I personally am not planning to work on that.
Okay, thank you. And are the judges ready?
Not yet. I'm sorry. I'm doing the second part. You know, this form is like consists of like multiple sections. Like I cannot go like all the way, all the things at the same time. I have to do first section done to go to the next section. So that makes things a little bit slower. This is the bottleneck, I think.
I have
Okay,
the
please
same
let me know
problem.
when you are ready because
I haven't even done the first one yet. I'm in the second one halfway.
Yeah.
okay, please let me
Because I
know when
didn't
you're
have
done.
this, yeah.
And while we are waiting, I think Ruby, Roya is still with us, right? There was a question for Roya.
Yes, can I answer that?
Yeah, sure.
Sure. So in that chart, the two other samples, CNT forest and the cover slips, were transferred to a new well plates for the pressable one just to test the surface cells on them. But the PS control was the polystyrene one, which was coated with gelatin. It was just in the well plate. I think the reason that the error bar is more compared to the other was it was different between the situation of the control. If I got your question right.
I think you did. Thank you for answering. Thanks.
You're welcome.
By the way, I'm done.
I'm almost done here. All right, done.
Okay, then let me introduce the presenter. So the presenter is Amanda Studinger from the Department of Chemistry, and her topic is energetic competition in the complexation affinity of paracetamol with other molecules. Amanda, are you ready?
Yep, all ready.
Okay, so I'll start now.
Okay. So this project models the intermolecular interactions of the active pharmaceutical ingredients. The active pharmaceutical ingredients are the actual drug molecules that interact with the body to produce a desired effect. These intermolecular interactions influence how the drug is manufactured and stored and how easily it's absorbed in the body, how it's transported within the body to the target receptor, and how it interacts with the environment in the water and in the soils. So after the APIs are manufactured, they undergo further processing so that they have the proper form and dosage to be administered in the body. And a growing area of research involves the use of co-crystal conformers with the API, and it helps to control the physical chemical properties of the API. So these co-crystals can help stabilize the API and increase the efficacy of the drug in the body. So for this project, we modeled a paracetamol, which is also known as acetaminophen, and oxalic acid complex. Okay, next slide. So the motivations for this work, we wanted to look at the driving forces of complexation and looking at the molecular geometries. We wanted to see how much energy was involved in this co-crystal forming. Was it ideal? Was it energetically favorable? There have been some examples of different drugs that a co-crystal was made and it worked, and then later a more energetically stable co-crystal got into the manufacturing system. And when it was administered to people, it just kind of passed right through them, and the drug was no longer working anymore. So we wanted to look at the complexation energy and the driving forces with complexation. We wanted to look at the competition between water and oxalic acid with the API to see how does the environment, both inside the body and out in like a soil system, influence and what drives that competition. Is it going to bind to oxalic acid, or is it going to bind to water? And then we wanted to look at the hydrogen bond distances, the differences in our modeling in a vacuum where it's just this molecule or these two molecules all by themselves in a complete vacuum versus what it would look like if it was in a solvated environment. And for this project, we used water as our solvent. Okay, next slide. So the computational methods that we used density functional theory, and we used 5 different functionals. When we were first working with this, looking at our results, we wondered if we wanted to make sure that our results were accurate and accurately representing our systems here. I'm sorry, I got distracted because my pictures aren't showing up on the slides, but that's okay. So we used 5 different functionals, and we used corrections for long range dispersion, which is that D3. So if you can go back, yeah, thank you. So we wanted to model all 5 functionals and see if there was a big difference, a notable difference, or if they all agreed with how our results were coming out. So to do this, we used a crystal structure from the CCDC database, and it provided us with the geometry of the paracetamol and oxalic acid. And we pulled the molecules straight from there and then modeled them to see how they behaved. And what we wanted to look at specifically were the hydrogen bonding sites with this complex. So you can see the one picture that did show up. You can see paracetamol is the larger molecule with the aromatic ring, and then oxalic acid is that smaller molecule next to it. And what we wanted to look at were the hydrogen bonds in between oxalic acid and paracetamol. So I highlighted just one of the examples, and I numbered the different positions around paracetamol because there are 4 different places on paracetamol where hydrogen bond can occur. So looking at this particular picture, this I labeled as position number 4, where oxalic acid is the hydrogen donor for that hydrogen bond on that hydroxyl group on the aromatic ring. Okay, next slide. So with density functional theory, just to give a little brief idea about it, it's using the Schrodinger equation to figure out the potential energy of the complex that we're looking at. With density functional theory, instead of using a wave function, like you see in that equation down at the bottom of the screen, it uses electron density. And then the Hamiltonian being used is a mini electron Hamiltonian. It covers the kinetic energy, the electron-electron interaction, and the potential energy that's present in the molecule and in the complex. And then when the calculation's finished, the ground state energy that is calculated is the ground state electron density. Okay, next slide. So again, like I mentioned, we use different functionals, B3 leap with corrections for what's called empirical dispersion. And 2 different kinds of corrections, just D3 or D3 with BJ. We used MO62X and omega B97XD. We use the same basis set for everything. So the basis set would be that electron density wave function in the Schrodinger equation. And we use the same basis set for everything. And then we corrected for any basis set superposition error. So if the any of the energy from oxalic acid overlapped with any of the paracetamol, we corrected for that overlap. This figure that you see on the right is showing that correction for the long range dispersion forces. And that's basically that idea of London dispersion forces, where there's that temporary dipole moment that happens within a molecule. And it can happen between molecules, it can happen within a molecule itself. So correcting for these long-range dispersion forces is a way to take into account those dispersion forces that are present in the molecule. Looking at the functionals MO6, that's something that's built into that functional. So we wanted to see, what are the results like if we just use M2O6, MO62X versus MO62X with corrections for empirical dispersion. Okay, next slide. So looking at the results, first thing we looked at, complexation energies, and this is just in vacuum right now, so not with the solvated environment. So what we're looking for is the lower energies, the more stable complex. And what you can see in this figure here, in this chart, looking at the different positions where the different hydrogen bonds are present, the most stable was at position 2, where the complexation energy was the greatest. Looking at the figures on the right, you can see this is what the results that we got from our calculations from what the complex looked like in vacuum in the crystal formation versus what they looked like in vacuum after they were relaxed, allowed to relax. We wanted to see, again, is there an energy barrier for actually forming in its complex? So right here, that first picture, that big change, that difference is with that nitrogen, hydrogen, and oxygen hydrogen bond happening. And we noticed a big change in the geometry with that oxalic acid when we relaxed that structure. Versus at position 2, which you see down at the bottom there wasn't that much change with oxalic acid when we relaxed it. Okay, next slide. So we wanted to compare the complexation affinities of oxalic acid versus water with PCA or paracetamol. Again, we're looking at what kind of competition is there, what are energetic barriers that would prevent PCA from binding to the target receptor because it's in an aqueous environment in the body, or what would drive it to form a complex in a soil system. So we looked at the differences in the hydrogen bonds. We wanted to see how they changed and how they varied. The idea being that the shorter the hydrogen bond is, the stronger the hydrogen bond is. So looking at the figure on the left, we're just looking at distances in angstroms. And again, you can see that at position 2, it has the shortest distance, and then position 4 has another shorter distance than 1 and 3. And I wish you could see the position so you knew what I was talking about. But in these areas, oxalic acid acts as a hydrogen donor. And on the PCA, it's just oxygen is the hydrogen bond acceptor. So what we noticed was with water and oxalic acid, they were both lower, but when comparing water versus oxalic acid, it would bind stronger to oxalic acid than to water. So that figure on the left is showing that oxalic acid prefers those positions 2 and 4 versus water. Looking at the figure on the right, we're looking at energy differences and comparing the energy of PCA and oxalic acid versus PCA and water. And again, we see that at positions 2 and 4, where oxalic acid is the hydrogen bond donor, there's a lower energy and a more stable complex. Okay, next slide.
1 minute left.
Thank you. So then we wanted to compare what it was like in a vacuum versus in an aqueous environment. Again, my figures didn't quite show out, but the circle around that molecule is to show kind of what happens when we model an aqueous environment. So what we're looking at is the differences, again, in hydrogen bond distances. And you can see that the hydrogen bonds are shorter in an aqueous environment than they are in vacuum. So that was both for oxalic acid and water, and PCA and oxalic acid. Okay, next slide. So to conclude, when we looked at all the 5 different functionals, again, we wanted to see how they behaved with this system. We saw that they were all in pretty good agreement with each other. The energy results varied by a little bit, but not much. So plus or minus 3 kilojoules per mole. Was that my time?
Yeah.
Okay.
You can quickly finish within a
Okay.
second.
All right. Thank you. The complexation actually is driven at positions 2 and 4. We saw that with the more stable energies, and where oxalic acid was the hydrogen bond donor. And then again, we noticed with differences in changing, a relaxed structure versus a crystallized structure, there were some changes in some positions, but it looks like when comparing the energies that the stability gain from making that hydrogen bond seems to outweigh any geometric strain from the complexes.
Okay, I think now judges can ask the questions.
Okay, thanks.
I jump quickly in because I have a really quick one. As a biologist, this was completely over my head. I'm sorry, they should have found a different judge for you. But can you give me just the biology version of what just happened? What's the take home for me when I go back to the lab and tell my student about what I just learned?
Yeah, so you think about biology, maybe the soil ecosystem might be a good piece of it to look at, right?
Yeah.
And when you're looking at an ecosystem, constantly balancing, this thing is here, this molecule is here. Will it stay in the soil system? Will it get washed through the soil system? Does it want to bind to the soil? Does it not? Right. And there's all these factors you have to take into account. What microbes are there? What plants, animals? So many things to take into account. So
Right.
what this is doing is just looking at one piece of it.
Okay.
Right. Comparing how do the hydrogen bonds work here, and what's more favored, one thing over the other. So as a biologist, right, you're looking at everything and trying to balance which one is going to outweigh the other one, right? And then what's the overall result? So what I'm looking at is I'm honing in at this one piece here, so that maybe when you're trying to balance it all later, you can weigh that in and say, well, no, I know that oxalic acid is a good makes good strong hydrogen bonds.
Okay, sounds good. Thank you so much.
Sure.
Yeah, so thank you for your follow-up, your kind of the summary in response to Dr. Warner. So yeah, it's just outside my field. This is very chemistry heavy work probably, and also computational chemistry work probably. So when you say, results among these, different functionals or in good agreement with each other, that each other means that like for the 5 functionals, like they are kind of showing similar kind of trend. Is that what you say in the first?
Yes. So for each complex, I did a calculation with that functional, and then I did it 5 times, and then compared the results that I had. And they were all very similar. Standard deviation was very small. How they varied, how the results varied, they were all very similar. So when I look at my results and I say, well, at what's my threshold for like my detection limit kind of a thing, it's 4 kilojoules per mole. That's where like I can say I'm accurate within that amount of energy. And looking at my functionals and seeing that they all gave similar answers that were within 3 kilojoules per mole, I would say that for all intents and purposes, they're the same.
I see. So whenever you see negative energy, that means that they are in very stable situation where they have some complexation. Is that how you interpret this data?
Yeah, the negative energy means that the molecule or the complex exists, right? There's more potential energy present than there is repulsive forces.
I see. And then this energy is calculated from this density functional theory. Is it how, or?
Yeah.
Right. Okay. Thank you so much. So, and then just like regarding this one, is this density functional theory the do you think that's the best way to show this complexation, or is there other well-used theory or methods to show the complexation happening?
Yeah, there are a lot, actually.
Really?
A lot of
Okay.
different ways to do it. Density functional theory is used because it's been shown to be a good approximation. Everything is an approximation. Once you throw more electrons into a system, you can use the Schrodinger equation for like hydrogen, it's great. And then once you throw more things into the equation, you can't do it anymore. And so you have to start approximating things. And so density functional theory is well used, and it's been shown to do a good job. But there are other models, and there are things like Hartree-Fock or molecular dynamics. So this is just one of those methods.
I see. Thank you. That's really good to know.
Okay. Please let me know when you are done with the scoring. And while judges are scoring, I'll open the platform for the general audience to ask questions. Please put your questions in the chat.
I'm done scoring. This is me, Thomas.
Okay, thank you. There's a question in the chat. Are there any other methods, basis sets that you are considering going forward with this project?
Going forward, I think we're going to stay with the method that we're using and explore more avenues. So bigger complexes, bigger pieces, and then not just looking at just the hydrogen bonds, but looking at the maybe the pi-pi stacking or the van der Waals forces that are present. So I think for now, we want to stay with what we have and build it larger to see if it still behaves the same way.
And any other questions? And I apologize for what happened with the Google Slides, especially
Okay,
for
things
Roya,
happen,
and
right?
it happened with Roya as well. So I apologize both, Roya and Amanda. I apologize both. And um, and
Yep,
I have,
I'm done.
yeah, okay, thank you. So thank you, judges, for your cooperation and help with the session. And I invite you all to go to the other sessions and check the other presentations as well. And
Yes.
and also I invite you all to attend the virtual award ceremony tomorrow, the GRC banquet ceremony tomorrow from 5 to 7 p.m. And link will be on the website gsg.mtu.edu/GRC. And if you have some other questions regarding or anything, please forward them to Sarada Chipkar, the research chair of GSG, gsg-research@mtu.edu, or to me, at gsg-prodev@mtu.edu. And thank you all for your cooperation, and thank you all for joining the session.
Thank you. Thank you for the presenters. Thank you for the moderation.
Yep, great job, everyone. Thank you.
Good to meet you, Dr. Twarner, too. Yeah.
You too.
Bye. Bye, guys.
Thank you.
Bye.